Large-scale genomic studies of non-model species have been challenging, time-consuming, and expensive, especially for non-human primates. Next-generation sequencing technologies provide an inexpensive way to generate high-resolution genome sequences of expressed genes. We describe the results of a large, parallel sequencing effort to generate sequences of expressed genes in the western North American pine, P. The results show that this pine has an extensive and well-established genetic base, and many of the identified molecular markers can be used to further study this pine and other non-model pines.
This article is titled Transcriptome Sequencing in an Ecologically Important Tree Species, Assembly, Annotation, and Marker Discovery and was authored by Benkman Craig W., Grahnen Johan A., Geist Katherine S., Parchman Thomas L. and Buerkle C. Alex.
We are article.tv. Article links can be found in the video description.